I was just reading the latest clinical data on mitochondrial function in neurons, and it changes how we should evaluate cellular energy decline. The brain consumes massive energy to survive. We often assume that cognitive slowdown is an inevitable consequence of aging. However, the mechanistic reality is that the brain's cellular engines may simply be running out of available fuel. By tracking the precise pathway of cellular energy depletion, we can identify exactly why neurons fail. This shifts our focus from general cognitive symptoms to specific local metabolic maintenance processes. Neurons are structurally unique, spanning immense distances compared to other cells. They demand constant ATP at distant synapses, yet primary power plants are located far away in the cell body. How does the neuron maintain energetic stability across these massive physical distances? When this transport and generation system slows down, the synapse faces a severe, highly localized cellular energy deficit. This local deficit is the true origin of synaptic silence. Before a neuron structurally decays, it undergoes a silent metabolic crisis that halts critical communication pathways between active brain cells. In the aging brain, we observe a gradual decline in processing speed and memory recall. This performance drop represents a macroscopic symptom of a macroscopic, essential cellular cell-level energetic bottleneck. As mitochondria age, they undergo structural damage, reducing their capacity to generate ATP. This decline in cellular energy output directly compromises the electrical signaling required for healthy, active cognitive processes. Simultaneously, dysfunctional mitochondria release high levels of reactive oxygen species. This oxidative stress directly damages the very proteins and lipids needed to maintain mitochondrial structural integrity over very long periods. Separate the mechanism from the outcome. The visible loss of cognitive function is merely the final result of this long-term, self-reinforcing cellular cycle of localized metabolic and harmful mitochondrial damage. The accumulation of oxidized membrane components reduces the efficiency of the mitochondrial electron transport chain. Consequently, the rate of cellular energy production falls even faster in the active local neuron. This creates an energetic threshold. Once ATP levels drop below a critical point, the cell can no longer support both basic survival pathways and complex synaptic transmission in brain networks. To understand this crisis, we must look at adenosine triphosphate, or ATP. It is the primary molecular currency that powers the essential active sodium-potassium pumps located in neuronal cellular membranes. Mitochondria exist as a dynamic, interconnected network within the cell. They constantly undergo fusion and fission to share resources and isolate damaged segments for degradation through specific cellular mitophagy pathways. Now, in highly active neurons, this network must extend deep into the axonal arborization. This distribution requires a highly complex transport system moving along our specialized active cellular microtubule tracts. Axonal transport is itself an energy-intensive process. Motor proteins consume ATP to move healthy mitochondria down the axon and return damaged ones to the central cell body for cellular recycling. Under normal conditions, this continuous transport maintains metabolic equilibrium. Active synapses receive fresh mitochondria, while old organelles are recycled through localized mitophagy degradation pathways within the central cell body soma. Synapses also rely on local protein synthesis to repair metabolic machinery. Without local mitochondrial support, the proteins required to sustain synaptic vesicles cannot be maintained by the active local neuron. As the organism ages, the efficiency of axonal transport motors declines. Mitochondria begin to accumulate damage in transit, stalled far from the recycling machinery located within the central cell body. This stagnation leaves distant synapses vulnerable. Without a continuous supply of functional mitochondria, The energy reserves at the local synapse cannot meet peak demands during periods of intense cognitive activity. The cascade begins when stalled mitochondria lose their membrane potential. Without this electrochemical gradient, they cannot import the essential proteins needed for basic cellular respiration and vital localized energy production. Instead of producing ATP, these damaged local organelles leak electrons directly to oxygen. This creates reactive superoxide radicals, initiating localized oxidative damage to surrounding synaptic structures and essential cellular membranes. This localized oxidative stress directly impairs the membrane proteins responsible for neurotransmitter release. Synaptic vesicles can no longer dock or fuse efficiently with the active cellular cell signaling membrane surface. Simultaneously, the lack of ATP slows the essential sodium-potassium ATPase pumps. The neuron struggles to restore its resting potential after firing, leading to immediate local signaling fatigue and communication breakdown. Follow the evidence boundary. We are looking at a mechanistic chain where mitochondrial decay leads to calcium imbalance, halting the electrical signals long before the active cell itself actually dies. Under healthy conditions, mitochondria act as calcium buffers during intense signaling. When they fail, intracellular calcium rises, triggering specific enzymatic pathways that degrade structural proteins within the local synapse itself. This structural degradation targets the microtubule network. As these internal tracks dissolve, axonal transport stops entirely, isolating the synapse from all essential somatic support and critical local metabolic resource replenishment. The isolated synapse is eventually pruned by glial cells. This loss of connectivity occurs not because the cell body died, but because the local energy grid collapsed at local synapses. The local collapse sends signals back to the soma. Retrograde transport of injury signals informs the central cell body that its distant connection is no longer functional under these conditions. So, the neuron systematically retracts its axon to conserve resources. This retraction is a survival strategy, reducing the active cell's energetic footprint at the cost of vital active synaptic connectivity. The debris from degenerating synapses triggers local microglial activation. These immune cells release multiple inflammatory cytokines, which further impair essential mitochondrial function in neighboring local neurons across the brain region. This inflammatory signaling accelerates the loss of membrane potential in adjacent mitochondria. The local cellular energy crisis spreads, creating a wider patch of metabolic dysfunction in the neural tissue network. As the patch grows, cognitive networks show measurable drops in local activity. The brain must reroute signals through less efficient pathways, consuming even more available energy from already strained systems. This systemic shift increases the overall metabolic load on remaining healthy neurons. They must fire more frequently, accelerating their own mitochondrial wear and tear in this vicious metabolic feedback loop. This final stage is the transition from localized synaptic dysfunction to widespread network failure. The aging brain's essential cognitive reserve is steadily depleted by this self-reinforcing chronic cellular energy deficit. To evaluate this model, we must establish its clear boundaries. Not all neuronal populations are equally sensitive to this metabolic pathway of mitochondrial degradation and subsequent local synaptic connection decline. For instance, short local interneurons with low firing rates demonstrate significant resistance to energy depletion. Their compact structure allows efficient diffusion of ATP without relying on distant axonal transport systems. Additionally, this metabolic pathway is primarily mapped in preclinical models. Directly measuring mitochondrial respiration in living human synapses remains a major technical limitation in our current clinical neuroscience research today. We must also distinguish between correlation and causation. While neuronal mitochondrial decay correlates with cognitive decline, it may coexist with other parallel aging mechanisms that directly affect synaptic health status. However, in conceptual models of accelerated aging, restoring mitochondrial respiration prevents vital synaptic loss. This provides a clear case where metabolic rescue effectively preserves structural integrity under specific laboratory conditions. This mechanism does not explain all forms of cognitive decline. It specifically describes the essential bioenergetic vulnerability of long projection neurons located in the aging human cerebral cortex and local hippocampus. By defining these boundaries, we clarify our main target. We are addressing a localized metabolic vulnerability, not a universal or uniform cellular decay process affecting every critical brain cell equally. This brings us back to the primary question of how the aging brain runs out of energy. The answer lies in localized, transport-driven, axonal, and synaptic essential mitochondrial failure pathways. Synapses do not simply wear out from use. They fade because the local mitochondrial engines can no longer replenish the molecular energy required for constant and efficient active chemical neurotransmission. By maintaining mitochondrial quality control, the active cell can theoretically prevent the accumulation of dysfunctional organelles that leak electrons and directly damage the synapse through localized chronic oxidative stress reactions. Mitophagy, the selective clearance of damaged mitochondria, is our primary essential cellular defense. When this clearance is active, the rate of critical synaptic degradation is significantly reduced in laboratory models. Simultaneously, mitochondrial biogenesis must generate new essential organelles to replace recycled ones. This balance between clearance and creation is the core of vital cellular metabolic stability in aging brain systems. Resolving this metabolic crisis requires protecting our transport machinery. Healthy microtubules ensure that damaged organelles return to the central cell body before causing severe oxidative damage to critical local synapses. When transport is preserved, active synapses retain their signaling capacity even in advanced age. The energetic bottleneck is cleared, allowing normal essential communication pathways to function without premature metabolic failure. Health span over lifespan. The essential objective is not to extend the lifespan of a dying neuron, but to preserve the vital energetic health of functional synapses in the aging brain. This bioenergetic model shifts our focus from structural preservation to functional support. Protecting the synapse means protecting the local mitochondrial engines that power its constant essential electrical and active signaling. In practice, we can monitor the physiological biomarkers that reflect cellular stress. While direct synaptic measurements are impossible, systemic metabolic health often correlates with overall general neural efficiency in humans. Supporting essential mitochondrial function involves managing systemic circadian cues, restricting daily energy intake cycles, and optimizing light exposure can support baseline cellular respiration pathways in healthy adults under normal conditions. Another vital consideration is pacing cognitive demand. Allowing periodic recovery periods during intense mental effort aligns with the essential rest and repair cycles of active neuronal mitochondria within our active human brain. What matters clinically is these protocols do not replace medical interventions. They represent lifestyle adaptations designed to support general metabolic pathways in our own clinical nervous system without medical prescription. When evaluating these concepts, tracking sleep quality and systemic glucose stability provides valuable indirect feedback. These markers reflect the general metabolic background of our organism under these studied lifestyle conditions. This discussion is solely educational and does not constitute individualized medical advice. Consult a qualified healthcare professional before modifying protocols, especially regarding metabolic markers and available cellular health evaluation options. By viewing cognitive maintenance through this bioenergetic lens, we can make better informed decisions. We focus on protecting the mitochondrial systems that sustain our complex, active, vital neural communication networks. Focusing on critical local cellular efficiency helps clear the noise of unproven solutions. We target the fundamental energy crisis that drives neuronal aging by optimizing our essential daily metabolic habits. We must evaluate crucial cellular energy decline by looking at the active local mitochondrial engines. Optimize the protocol, protect your brain, and I'll see you in the next dataset.